Great. So I'm going to switch gears just a little bit here. So the first thing I need to tell you is I'm from an academic lab. I'm not here to talk about my business plan. What I am here to talk to you about is how we're using LGC's service to help us go from sequence variation to functional characterization of loci in tomato. So I think that's the next thing I need to tell you is that my group works on tomato, both from a practical point. We do create inbred lines that we license to industry. We occasionally release hybrids. So we have a very practical bent to what we do. At the same time, we're an academic program, and so we're meant to be doing discovery research that contributes to knowledge. This slide summarizes what I've been trying to do for the last 2 or 3 years, and that is to distill information from publicly available genome sequences to address some concerns or improvements that we would like to make. So basically the situation is this. In 2012, the first reference genome for tomato became available. In the subsequent 4 years, we gained close to 450 genomes. So the question is, how do we use this information to push our breeding goals forward? And so what I'm going to talk to you about is really just 2 examples from our program. The first involves high beta-carotene tomatoes. So these would be tomatoes that have a different color and are extremely high in beta-carotene. I'm going to give you an example of how we've done this in processing tomato backgrounds, and then I'm going to just talk briefly about a master's student project in a grape cherry tomato background. And then I'm going to give a parallel example where instead of looking at a specific gene, we're looking at QTL for bacterial spot resistance. And I'm going to be talking about how we're using the LGC resources to help us push both marker-assisted selection but also background genome selection. Okay. So in order to achieve this, we need a pipeline, and this is how I think about our pipeline. So we have sequence data that goes in, and what needs to come out are alignments, either sequence, protein, for the structural gene, or in a couple of cases, promoter alignments, or 5' sequence alignments to be more accurate. And so even though we have 450 genomes available, the truth is that 85 of those are of really high quality and are suitable for this analysis. And then we've added 7 of our own so that we have a context. So the pipeline was put together for us by an undergraduate student named Gabriel Aboud, and I've put his picture here so you can see him. And basically what we're doing is retrieving, aligning, and clustering 5' sequence, gene sequence. We're converting the genes to proteins, or 3' sequence for any candidate gene that we want to study. And so the first example I'm going to give you is a result of a master's student project. So this is Caleb Orchard, who now works for East West Seed in Chiang Mai, Thailand. Caleb was interested in this idea of improving nutritional quality by improving the beta-carotene content. And we knew that there were alleles of this fruit-specific beta-cyclase gene that could lead to either high lycopene or high beta-carotene. And a lot of this work had been done over 50 years ago, and then the gene was cloned by Hirschberger's group. So we had some information that we could work off of. And so Caleb's first question was to address what was the allelic diversity for this gene, which is known as beta. And then to develop genetic resources with different alleles for the functional evaluation. So the first thing that Caleb did is he went to seed catalogs, he went to National Plant Germplasm System, the Tomato Genetic Resource Center in Davis, and anything that had a color that might suggest it was high in beta-carotene, he requested. We have a cooperator, Steve Schwartz, on main campus, who's one of the world's best carotenoid chemists, so we use him to help us phenotypically evaluate this material. And then anything that came through this analysis with high beta-carotene, Caleb sequenced using Sanger sequencing approach. About the time he finished that, the 150 genome project dumped a bunch of data into our lab, and so Caleb could ask this question: what is the allelic variation available? And so this tree is basically showing natural variation in the 5' region of this fruit-specific beta-cyclase gene, okay? And so what we found is that there were 25 or more alleles within the structural gene and about the same number out in the 5' region, the promoter region. Okay, so quite a bit of diversity. And another way to look at this diversity is shown in this slide. So to the right-hand side is a cluster of sequences that come from green-fruited wild relatives. These are cross-compatible. We can make crosses and integress this segment into a cultivated background. And when we do this, we get an orange tomato. And then over to the left-hand side are the wild tomatoes that come from the Galapagos Islands. This would be Solanum cheesemanii and Solanum galapagense. Those fruit are orange and high in beta-carotene. So we have a couple of examples of genes that we know are going to give us high beta-carotene, and what we wanted to do was assess the function of these different alleles in a uniform background. So I'm just going to take a little bit of divergence, and then I'll get us back on track. Now, with the sequencing and phenotyping that Caleb did, we were able to use an association approach to ask, given all the sequence variation, and the sequence variation in 1,500 bases are represented by these black bars, given all the sequence variation, which are likely to be functional? And there were two SNPs that came out of this association analysis that were present in all high beta-carotene promoters, and those are shown in this alignment here. So we think we were able to get to actual functional SNPs, and we've got a project going on in which we're actually trying to prove the function of these SNPs at imparting high beta-carotene. Now back to Caleb's mission. He was interested in evaluating specific alleles in a uniform genetic background, and so his approach was to use a backcrossing strategy, but he wanted to accelerate it. So for those of you that aren't breeders, the idea of backcrossing is that each subsequent generation of backcrossing, you eliminate half of the donor genome. As long as you're selecting for the trait of interest, you bring it along, and after several generations, you arrive at a state which you have essentially near isogenic material. That's great if you have a long time. If it's a short amount of time, you need to figure out how to speed this up. And that's where background genome selection comes in. And so what I'm showing you in this slide, on the top left, you have a flat full of tomato seedlings. We extract DNA out of those. In our lab, we just use an insertion deletion mutation to identify the heterozygotes. Those heterozygotes, we move on to the next step, and for us, the next step means we send the DNA to LGC. We tell them which SNPs we want to use. In our case, we're using SNPs spread across the genome, and so the bar graph is showing you black as the donor genome, white as the recurrent parent genome. What we're getting in that first backcross is a distribution, and I'll show you this in a little bit. And what we're using the markers to do is to identify individuals that have more recurrent parent genome than the average. Those we advance to the next generation and evaluate after selfing. Okay, so let me just talk a little bit about the resources. And this is another point where I can thank Martin for his work. So as part of the SoulCap project, we worked with Illumina to develop an array with almost 8,000 SNPs that were optimized for use in cultivated backgrounds. Both Martin and Matilda Kos cooperated with us to add content to this and develop a really robust genotyping array. As Martin mentioned, though, there are many cases where you simply don't need 7,000 markers, and most tomato breeding populations are a good example of that. We simply don't have enough recombination to make every marker unique, right? And so why pay for data points that you don't need? And that's where LGC came into the picture. So LGC worked with us to make the SoulCap SNPs available as CASP assays. And so for Caleb's project, we could just pick and choose the SNPs we wanted. And I'm just going to show you a little bit about the interface. So I'm just using screenshots here, but there's a page that's specific for the tomato assays. If you know the SolCap SNP ID, you can type it in and add it to your cart, essentially. Okay. I think I'm going to just go through this really quickly. The same pages, you can pull out PDFs that describe the CASP genotyping technology if you're not familiar with it. You can purchase this as reagents and do it in your own lab, or you can use LGC as a service provider where you send DNA, or even tissue for that matter, and then get your data back. So the CASP assay is essentially a competitive allele-specific primer extension assay. One of the things that I've been quite happy about in using them as a service lab, I went back through data, and we have well over 10,000 data points that we've worked with through them, and we're getting over 98.5% data return. Okay, so very little missing data in this process. And I think the other thing I should say is we do all the DNA preps in our own lab. We're doing them 96 well plates at a time. We process 4 plates in a morning, so we're not quite as efficient as the talk that you heard earlier, but still fairly efficient. It's a CTAB protocol, and we get more than enough DNA for our use, and it's high enough quality that we can sequence off of it, and we can genotype off of it in a number of platforms. There's another piece of information I need to throw into the mix here, and this is something that sort of reinforces what Martin was telling us earlier. And that is that by using SNPs that are on common platforms and common SNPs, we can begin to pull together large data sets. And so this paper that I'm showing you here was a collaboration with Jose Blanca's group in Valencia. And the supplemental tables have genotypic data for 1,000 tomato accessions with the SolCap SNPs. And so when it comes time to select which SNPs you want to use in a project, you can go to this data and actually pre-screen. You've got 1,000 accessions. So maybe your own parents are there, but if they're not there, you can look at pedigree information or historical use information and create sets of SNPs that are very likely to be polymorphic in your populations without having to screen the parents across all the information. And this is all publicly available information. Okay, and so now I'm going to jump back into Caleb's project, and again, what I'm showing here, let's just concentrate on the left-hand side. You have a distribution that represents the Backcross 1. The proportion of recurrent parent genome is on the x-axis, and the number of progeny is on the y. And what you can see is you get a distribution that approaches normal. The mean is right what Mendel tells us it should be at 75%. But we have individuals in this population that are a full backcross ahead, right? If we take those and make the next backcross and repeat this process, in 2 generations of crossing, we have individuals that are approaching near isogenic. And we self those, select the genotypes we're interested in, and guess what? It's time for functional analysis. And so as a result of Caleb's work, this is what we see. We can identify alleles that are higher in beta-carotene. The Pinelli allele was the one that was cloned by Hirschberger's group, but you can see that Habericades and the Galapagos give us alleles that give us much more beta-carotene. So we have an allelic series here that we can choose from to modulate the amount of beta-carotene in an inbred processing tomato line. So the other thing I can show you here is that if we look at the expression here, that expression pretty much mirrors what we see with beta-carotene levels. Okay, and so these, the high beta-carotene is conferred by SNPs in the promoter that modulate the transcription of the fruit-specific beta cyclase. Okay, so the second example I'm going to go through really quickly here had another student that came into the lab who was more interested in quality, and she wanted to work with this high beta-carotene material in a grape tomato background. Okay, and so Eka basically took the same approach, and what she did is she worked with a set of SNPs that had been optimized for use in fresh market backgrounds, and this was the first set of SNPs that LGC made available publicly. They now make all essentially 8,000 SNPs available, but there is an optimized set, optimized based on polymorphic information content and optimized based on distribution across the genome. And so ECHA selected SNPs from across the genome and essentially repeated the process that Caleb went through and identified individuals that had much higher percentage of recurrent parent genome. And then what she did is she evaluated these for quality using a trained panel. And what she, in this process, what she did is she identified a, you could call it allele or a promoter. In this case, what we're really looking at is linkage drag around that high beta carotene gene. But one of these sources gave us much higher sweetness, tomato-like flavor, and other organolepto qualities that are preferred by consumers. And so, again, what the point is that using the SNP resources, we can very quickly do this background genome selection and evaluate material in a uniform genetic background in order to assess which alleles are better and which we may want to discard. And the photos there of ECHA are just to give you a feel for her personality. So final project I'm going to go through really quickly here since I'm down to about 3 minutes. Has to do with the disease resistance project. So bacterial spot caused by 4 different species of Xanthomonas is a major problem for us in the Midwest, and it's a problem anywhere where tomatoes are grown under a humid environment. We've been working with this disease for some time. We have resistance selections that have come out of different sources of resistance. And one of the things that's been showing up over and over again is that there's a QTL on chromosome 11 that we've identified with 3 different resistance sources. And so what we wanted to do was evaluate whether this chromosome 11 QTL was equally as effective regardless of which source it came out of, or whether there was one that we should focus on over another. And this is work that was done by Eduardo Bernal in my group. And so again, he took a backcross strategy using QTL from Hawaii 7998, from a wild cherry tomato, this PI 114490, and then from a Pimpinellifolium. And so he repeated this basic strategy. This shows the distribution of markers. The red bars represent the region on chromosome 11 that he was selecting for, where the QTL is. The black bars represent the background markers that he identified and chose. Okay. Here's the distribution after the first backcross. Okay, so we're looking at the recurrent parent background, and we're looking at the number of individuals, and then the fruit from his selections are there. So you see that in a single backcross, he was able to identify material that was equivalent to, say, a BC4 and had fruit size that approached our recurrent parent. And then when he evaluated this in the field, in separate trials under infection, what he found was that in certain cases, and so like the top left where we have infection by Xanthomonas gardneri, that the allele from the Pimpinellifolium in green there is probably the best, although it's not statistically better than any of the others. Over all 3 species, the Hawaii 7998 allele looks to be the most effective at giving us resistance. And we now know after sequencing some pedigrees that these QTL really are different, but now we can prove it, and all in the context of a master's thesis, so in simple 2-year project. And so that concludes what I wanted to say. I do need to acknowledge that I've had many collaborators and many funding sources on this. So if there are any questions, I'm happy to take them, but given the past 3 talks, I expect that there will be no questions and we can move on to the next talk. Glendon. Okay, there is one hand raised. So.
What size population were you genotyping for your Backcross 1?
Okay, good question. So what size population were we genotyping for our Backcross 1? So larger than we needed to. So you can very easily get, you can achieve one generation ahead with a population size of about 96. Okay, and that's, if you go to the literature, especially work coming out of Fritz's group in Germany, that's what theory would tell you as well. We tended to work with populations that were 3 or 4 times that size, and we probably won't do that anymore. Okay.